Hello and welcome to what is going to be a series of videos over the use of advanced microscopy and some of the applications and techniques and kind of the fundamentals needed to be able to conduct research using a microscopy approach. So I want to start with just a brief history of microscopy. So the first compound microscope is largely credited to Hans and Zacharias Janssen in 1590. Anton von Leeuwenhoek was the first to observe bacteria. I'm sure in your biology class you heard about the animalcules discovery in pond water. Charles Hall invents achromatic lenses. So these early microscopes, these are all candle-based microscopes. So their light source is actually a camera that they put underneath the sample that illuminated through the sample. They are largely using just one single lens, and they were not a compound microscope like what we're used to today. In 1830, Joseph Jackson Lister discovered that you can use multiple weak lenses in combination with each other to generate more magnification without some of the conflicts that come from just doing it with a single lens. And then we finally start to move into kind of what I consider the modern era of microscopy. After we are able to discover the UV radiation emits blue light, and we can start to use different colors of light to illuminate samples. And then in 1911, that develops into the first fluorescent microscope, and now we're truly into the modern age of microscopy and microscopy techniques that we're familiar with today. This was probably the single greatest discovery in all of microscopy, the discovery and purification of wild-type GFP in jellyfish. This discovery changed the game completely and made it so that we can not only image things easier, but we can also genetically encode things to be fluorescent and allow us to look at it. We're going to go over GFP and fluorophores in more detail later, but this is really kind of the birth of the modern era of microscopy. And then in 1986, the first digital microscope was made in Japan, which led us truly into the modern era of microscopy, where we're no longer just looking at images through the eyepieces and taking pictures with a film-based camera, but we're now looking at digital images that are quantitative in their nature, and we can actually start to assess them for data. So why do biochemists and biologists in general rely on microscopy to conduct their research? Well, the first thing and the most important thing is that microscopy allows us to get cellular context to biochemical processes, where if you run something like a protein gel, a Western blot, the traditional biochemical analyses, you're not going to get much cellular context out of it. Yeah, you can purify a protein and know that there's a protein there, but we don't know if that protein was associated to the ER or to the nucleus or to the cell cortex, or if it just floats in the cytoplasm. So by using microscopy and specific labels, we're able to give compartmentalization context of these biochemical processes. And we can look at distinct localization of biochemical events. So we can see things like microtubules and where they're at. We can look at proteins associated to certain structures within the cell. And then we can also characterize biochemical concentrations as they exist in the cell and where they exist in the cell. So if you have a protein that you think is highly cytoplasmic, we can tag it with a fluorophore, and we can see how much of it is abundant at any given time and how those levels might change based on genetic expressions. And then the coolest thing, I think, is we can manipulate biochemical compounds using the microscope. So we're going to get into a little bit later photoclick chemistry and photomanipulation of molecules using photon excitations, which is super cool stuff that we're going to get into, and it's becoming a very powerful technique in the field of microscopy and in the field of biology in general. So in any given microscopy course, there's going to be some kind of notable techniques that almost everyone will teach you and you'll learn in microscopy. The first being phase contrast, so that's using white light to illuminate things so that you can see them with more contrast. Confocal imaging, which is kind of the modern techniques that people apply to see really specific images, where we're using a pinhole to get rid of out of focus light, which we're going to get into. Fluorescent imaging, which is often used in combination of confocal imaging and other techniques as well. And then finally fret and frap, which I'm not going to talk about in this video specifically, but we're going to talk about it later, which is where we're looking at localization events of molecules using fluorescent probes. So to start with, there's kind of three tenets of microscopy. The first being magnification, so we're trying to make an object appear larger than it is. Contrast, which is by definition differ strikingly in appearance, which isn't very helpful really when we think about it, but we're going to get into why does that actually make sense and how do we make things differ strikingly in appearance. And then finally, probably the most important aspect of microscopy and the least utilized and understood is resolution. So the ability to determine the distance between two different objects, which we're going to get into resolving power and why that matters so much more, even more so in my opinion than contrast and magnification. So the first tenet, magnification. So I'm showing you just a very simple formula for magnification here, and I don't want you to get too lost in the weeds on the math. One thing to remember about microscopy is it's the tool of biologists, but it's really governed by physics, and that's what this image here is meant to demonstrate. These are laws of physics that are being applied to the way light behaves when they pass through a substance. But for our purposes, we don't really care what the physics says, we care about what it can do for us. I really like this image over here, right? So this is this deer as it would appear if you were just to look at it with the naked eye, no binoculars aiding you. And then you take a set of binoculars, you put them up to your eyes, and the deer is some fold change bigger in appearance to your eye after using the binoculars. We do the same process in microscopy. We're using a series of lenses to achieve a greater magnification of a very small object. So bacteria, cells, all sorts of fun stuff we can look at under the microscope. One thing that we should remember though in magnification is that magnification is determined by the lenses that you are using. So you're limited by the lenses you have available and how those lenses are interacting with the light. Okay, second tenet, contrast. So we're trying to make things appear strikingly different, right? Well, the way we can imagine this is through what's called bit depth, and the way bit depth is applied when we're using a digital microscope approach, right? So we're setting an opposition to show differences. So if we start here on the left, we have what's called a 2-bit image. What a bit depth is the separation between white and black. So you can think of white as 0, black as 1. And then we get shades of gray in between. And so the more bit depth that you have, the more shades of gray you'll have in between that 0 and 1 binary white or black. We can get all the way up to a 10-bit image. And at the 10-bit image, you can see that it's a very smooth gradient and there's very little difference between a lot of these shades of gray. Well, if we want to increase our contrast, one way that we can do that is by reducing the amount of bit depth that we have so that these shades of gray get binned more strikingly different in appearance. So you can tell the difference between this shade of gray and this shade of gray will be more dramatic than like this shade of gray and this shade of gray here. So by using our bit depth and our gray levels, we're able to set more contrast. The other way that we can do that is through manipulation of light, which is called phase contrast, which we're going to get into in a little bit here. One thing to remember is microscopes only see in black and white. There is no coloration of a microscope. Any microscope image that you see that is in color has been digitally modified to assign color to gray values. So you don't actually see in color with microscopes. They only ever see in black and white. All right, third tenet, resolution. Like I said, this is probably the most important aspect of microscopy that is by far the least utilized and least understood, but is probably the most important thing you're going to use. So resolution is the ability to tell distance between two objects. So this schematic on the bottom left here shows this really well. So when light comes through an objective, you get what's called the airy disk, which is the refraction pattern of light around a focal point. Well, what we care about in this schematic is the solid dot, right? This is our in focus best light that we can get. And you can see that in this fully resolved image, you can see there's one clear sphere here, a separation between these two spheres, and then the second sphere, So these are fully resolved. I can tell that there are two objects that I'm looking at. And this just resolved image, again, looking at the spheres in the middle, we can tell that there are two spheres there, but we do not see the difference, the distance in between them, right? So we can't truly say that these are two completely separate objects, but we can apply what we know about what we're looking at to infer, yes, there are two different spheres here, and they are likely different. And then an unresolved image will look like this. There are two spheres here, but we do not have sufficient resolution to be able to see the difference in these two objects and see a distance in between them. And so that becomes really important in microscopy when you're looking at things on the subcellular level. If you're looking at two proteins and you're interested in the difference between two proteins, they're going to be very small and they're likely going to be very close together. So you need a sufficient resolution to tell the difference between those two proteins that you're looking at. Otherwise, they're just going to appear as one solid object and you'll have no idea what those things are. Now, resolution is governed, by physics. Microscopes run on physics but are applied by biologists. The numerical aperture is what determines your resolution. Now, I don't want you to get lost in the math on the numerical aperture stuff. That's not our point. But you should know that the numerical aperture will differ between the different lenses that you're using. So n is the refractive rate between the specimen and the objective lens. So really high numerical aperture lenses are going to use oil immersion. And what that oil immersion is doing is it's making a consistent refractive gradient between the glass of the objective, the media that the light is passing through, which is your oil, the glass cover slip, and then your sample. Air has a different refractive index than oil does. So if you're using an air objective, you're actually going to lose some of your light and some of your resolution to diffraction as it collides with air molecules in between your objective and your cover slip. The objective angle is a function of the objective that you're using. You can't really modulate or change that. But when you pick up an objective, there's going to be some things on there that you should know about. One of them is your magnification. That will always be listed. A general rule of thumb, but not always true, is that higher objective lenses are going to have a better numerical aperture than a lower power objective. And that's due to the objective aperture angle of higher power objectives versus a low power objective. The second thing is going to be the numerical aperture, and it's going to be a number. An example of a really high numerical aperture lens is like a 1.4 numerical aperture versus like a 4x air objective, which is going to have around a 0.6 of numerical aperture. So in general, when it comes to numerical apertures, bigger is better. So if you're when you go to look at what you want to image, you need to take into consideration, do I need magnification more or do I need resolution more? And more often than not, what you really need is resolution as opposed to magnification. So it's really important to consider these things in conjunction with each other. So to get started on the actual functionality of a microscope, we need to talk about the light path a little bit. This is a very basic schematic of a light path that I drew up. So we're going to start with some sort of a light source. That can be white light, like from a halogen bulb, or it can be a laser, which we'll talk about lasers and why we might use them a little bit when we get to fluorescence. But lasers are going to use single wavelengths of light, where a white light source is going to have a multiple spectrum emission. And that becomes relevant when we get into fluorescence and excitation and emission of fluorophores. So you're going to start with some light source. That light source is going to shine into the microscope. It's going to go through a filter set, which is going to specify the wavelength of light that you care about. So in this schematic, again, we're starting with a white light illumination source, but we have filtered out all but the blue light. The blue light's going to hit what is called a dichroic mirror. The dichroic mirrors are designed to only let one wavelength of light bounce off. So in this case, if we're using like a 405 wavelength dichroic, only the blue light is bouncing off of it and getting sent up through our objective. Our objective then focuses on the sample. The sample is going to bounce it back, but now it's being bounced back as a green light, which can then pass through the dichroic because it's not in that 405 wavelength that the dichroic is designed to bounce. It's going to go through a pinhole aperture of some kind if it's confocal imaging, If it's not confocal imaging, this likely won't be here. Or you might have additional filter sets that filter this light as well, which will then go to our eyepiece or our camera, and then we're going to generate an image. So in fluorescence, the reason that fluorescence microscopy works, as I was alluding to in the last slide, is this concept called the Stokes shift. Now the Stokes shift is a change in wavelength of the light that you're using when it hits a fluorophore. So going back to our light path just a little bit, when we have an excitation light, we go through a beam splitter or a dichroic. It depends on what type of microscope you're using. That's going to come up to our objective, which is going to focus it on our specimen. Specimen's going to kick it back down. It's going to come back through again, through this lens that's a filter lens, through a pinhole to our detector. So if we're using a fluorophore that is specific to 405 blue light, what's going to happen is that light's going to come through. It's going to hit this dichroic or beam splitter. It's going to be bounced, reflected off that dichroic up through the objective and onto the specimen. The reason that we can get specificity out of our fluorophores is because when it hits that specimen, it's going to undergo what's called a Stokes shift. So over here we have this wavelength diagram. We have some intensity of power, right? Intensity is just a power. It's a brightness of the fluorophore you're looking at. It is absorbed by the fluorophore at this wavelength of light. We'll call this 405. And then when that photon is absorbed by the fluorophore, it undergoes what's called a Stokes shift, where it changes the wavelength of the light to a new wavelength. So here we're going to 488, which is our green channel. So light came in blue, underwent a Stokes shift, and got spit back out as green. And so now that we're in this different wavelength, we're able to pass through this dichroic, and we're able to go back to our detector or our eyepiece. So the only reason fluorescence microscopy works at all is because of this concept of photon absorption by your molecule and emission at a different wavelength. So now we're going to get a little bit into confocal microscopes, because likely if you're doing fluorescent microscopy, you're most likely using a confocal microscope. So we have all the same components that we had in our light path back here, right? We still have a beam, we still have a dichroic or beam splitter, we still have an objective, we still have a specimen, but we're adding in this component here, the pinhole, which on a LSM, a laser scanning microscope, is going to be a single pinhole with a changeable diameter, or on a spinning disc confocal, a series of pinholes that are going to, and what the purpose of these pinholes are is to get rid of out of focus light. So going back over here, we're using in conjunction with a wavelength. So we have our dichroics and everything, which is being shown here. We have these microarrays on a spinning disk. This is an example of a spinning disk confocal. And you can see that the disks are studded along this rotating platform here. And they're 50 micron pinhole arrays. And as light comes to pass through this, so it comes through our specimen, you can see that it starts out not completely focused. We have a wider beam here. And then it hits this spinning disk. This spinning disk is synchronized with the focal plane of what you're looking at. And so only the only the focused light is able to pass through the pinhole, through to the dichroic, through the micro lens array, and then to the filter set into your detector. And so what this does is prevent you from seeing out of focus light above and below the focal plane. Like, light can be bounced by many different things, not just your specimen, and that introduces noise into your sample. So what we want to do is reduce that noise and get rid of that light that's not relevant to the thing that we're looking at. We do that through the pinhole's spinning disk. And the reason this is really important is now we have X and Y resolution, right? We can see things on a flat plane. So if we're looking at our specimen here, we have X and Y resolution, but we also want to be able to convey depth of our samples. It's called Z-axis. And so we want to be able to see things in our Z as well. We want to know if a nucleus is not flat. A nucleus is a sphere that has a certain depth to it. By using confocal microscopy, we're able to get specificity of our focal plane at different depths in our sample. so that we can then see things in not only X and Y, but in Z axis as well. And if you take a sampling size that allows it so you get perfect alignment of your Z axes, we can see things in 3 dimensions, which is really powerful because biology happens in 3 dimensions. Very rarely are things flat when we want to look at them. Okay, so the first kind of major technique that is used is called phase contrast microscopy. And what this is going to do is you're going to use polarization of light by sending your white light source through a slit. In most modern microscopes, it's a phase contrast ring, which is a series of slits. But you're sending light through a slit to cause different wavelength patterning of the light coming through. And now this difference in the way the light is coming through the slit is going to cause you to have really high contrast between bright objects and dark objects. So again, we want to achieve contrast by making things strikingly different in appearance, and we've accomplished that here. So they're looking at HeLa cells spread out on a glass cover slip, and you can see we have a really nice edge of the cell. What phase contrast is really good at is seeing the outlines of things. So we have a nice outline of a cell here. We have a nucleus inside of the cell. We have some dark studded things that we don't really know what they are. Could be ribosomes, could be a lot of different things. So you don't get a lot of detailed information, but going back to our why do we use microscopy, we are able to get a compartmentalization in cellular context when we add other imaging modalities in on top of phase contrast. Phase contrast is also really useful if you're just trying to look at something quickly and you don't want to have to, go scan through everything on your slide and a really high objective. You can usually just look real quick at your stuff and see things easily. It's also really useful for things like wound healing assays, where you don't care about much of intercellular mechanisms, but you want to see how the cells function as a collective. Phase contrast can be a really powerful tool for looking at cells in bulk. Okay, and so now we're going to look at confocal microscopy using a fluorescent probe. So what I've done here is I've taken images of 2 different fluorescent channels. On the left, we have a mitochondria marker that is specific to 556 wavelength of light. And on the right, we have actin stained with a green fluorescent molecule. And then I'm using a confocal microscope to take a single section through the middle of these cells to be able to see them in a 3-dimensional context as well. And I'm seeing intracellular mechanisms of the cell. And what you can see is we get a really dark background. We get a really bright light. So again, we've achieved a really high, strikingly difference in appearance contrast methodology by using a fluorescent protein. And then those filter sets I was talking about where the dichroic is bouncing certain wavelengths of light. Our fluorophore doesn't exist outside of the cell because these are really well prepared cells. And so we have no background noise now because we've filtered all the ambient light out and we're specific to a single wavelength of light inside of the cell. So you can start to get really detailed images of cellular processes by using confocal in conjunction with the fluorophore. Most modern microscope images that you're going to see are going to be fluorescent molecules in confocal modalities. So now that I've introduced the idea of what you can do with a fluorescent molecule, I want to explain a little bit of how fluorescent molecules work. All right, so you want to make a glow-in-the-dark kitten. So there's a couple aspects of all fluorescent molecules, no matter what wavelength of light they're specific to, that you should be aware of. They are almost all this beta barrel structure, and inside that beta barrel there's an alpha helical structure that holds in place a chromophore. And it's this chromophore that is going to actually absorb a photon, undergo a Stokes shift, and emit a photon inside of that beta barrel. This is the functional business end of this molecule. And we can do this for multiple different wavelengths of light, which I'm going to show you later. But the most powerful aspect, I said this is probably the most revolutionary thing to have ever happened in microscopy. The reason that this is so powerful is, one, we can start to get specificity on what we're imaging, If I want to see one specific protein, I can label it with a fluorophore, and I can look and only see that color of light as I demonstrated on the previous slide. But also, we can genetically encode fluorophores. So CRISPR-Cas9 has been a second major revolution in not only imaging biology, but biology as a whole. We can CRISPR in fluorophores onto genes of interest that then, when that gene of interest is turned on, will append this fluorescent molecule to that protein that will be expressed in the cell, and we can then look at it. So if you're interested in looking at temporal resolution of certain proteins, this is a really great way to see it. Right? If a protein is only expressed in interphase, we can see it expressed in interphase by conjugating it to a fluorescent molecule, and then we're able to know it's only active during interphase, but it goes away during mitosis. So the reaction mechanism of fluorescent protein, we're going to get a little bit into the chemistry here. So again, we have a beta barrel, and inside that beta barrel is a chromophore. So I'm looking at the GFP molecule. So that chromophore, if we blow that out, is composed of a tyrene, a glycine, and a serine residue. And we have another side chain over here that's interacting as well. But the business end of this is that these three amino acids here are going to undergo a cyclization reaction. And so what we're going to have happen here is this nitrogen is going to bond onto this partial positive of this carbon here, leaving us with an OH group on this where this carboxyl was. And now we have cyclized the amine terminus of this tyrosine and serine molecules and made this cyclization. It's the cyclization that actually allows fluorescence to function. But we also need to get rid of this OH group, so we have a dehydration reaction, which is going to result in the leaving of water. And so once we've left water, now we have a fully mature and ready to go fluorescent protein. And this matured fluorescent protein can exist in multiple conformational states, which is why you get two different absorbance. And this different absorbance is based on if this terminal oxygen here on the tyrosine is protonated or not. So in the protonated form, where it's a hydroxyl group, you're going to have an absorbance of 397 nanometers, which again, that absorbance nanometer spectra is referring to a wavelength of light. So we're absorbing at a wavelength of light of 397 nanometers. If we deprotonate that, we're going to have an absorbance of 475 nanometers. So this version of GFP is what is most commonly used. You're most likely going to see this, but due to stochastic conditions in the conditions of your media, you might be absorbing at 397 or at 475. So your media does come into play on the ability of your fluorescent molecule to be fluorescent. Now, at physiological pHs, you're most likely going to be in this deprotonated form. But if you're ever wondering why you're getting so much off-peak excitation is what that's called, it likely means that you have protonation differences along your actual chromophore here. And then this is the functional part of a Stokes shift. So a photon is going to hit this structure, this cyclized structure here. It's going to be absorbed and resonate throughout that structure, and then it's going to be emitted at a different wavelength after it's undergone a wavelength transformation due to the energetics of the light that entered that fluorophore. So this right here is what makes it so that Stokes shift is possible and we can see things. So I mentioned that not all fluorophores are built the same and that we have different wavelengths of light that can be absorbed by different molecules. So what this graph here is showing is that these different molecules, based on their structure, are going to absorb things at different wavelengths of light. So this kind of small cyclized protein here is going to absorb the UV spectra 320 nanometers. But if you look at this backbone here, this cyclized component flanked by these two benzene rings, and then we append on some more side groups, we've changed our absorbance to 365 nanometers now. But we still have this same core structure, and it's this cyclized nitrogenous ring that's actually doing the absorption. And then we go down here to this one. Now we have a different architecture, but we still have our cyclized ring, and we're absorbing light at 405 nanometers. And then you go to this beast of a molecule, right? We've got a whole lot of stuff going on here, but we still have a cyclized nitrogenous ring that is absorbing at 700 nanometers now. And what you can see is because of these different absorbance spectras, we can use different colors of light to look at specific proteins. So if I want to look at two proteins at the same time, I can use a fluorophore that absorbs at 405 nanometers and a fluorophore that absorbs at 566 nanometers, and I'll be able to see different proteins based on their absorption patterns of light. So this is probably one of the coolest and this is kind of the bleeding cutting edge of chemistry using light right now. It's called photoclick chemistry. The reason it's called click chemistry is things seem to just click into place. But I'm going to give you the basics of photoclick chemistry and how it works. So we start with some sort of functional motif, right? This can be an antibody, this can be a fluorophore, this can be a protein. A functional motif can be anything. And onto that functional motif, we're going to add a molecular plug-in, right? So that's this benzene with this NO2 and OH group. And then we're going to take that molecular plug-in, we're going to subject it to UV radiation, UV light, which is going to cause a water to leave. And we're going to end up with a functional form now, where we have a nitrogen bonded to an oxygen with this hydrogen here and this carbonyl carbon over here. And we can take this functional molecular plug and we can conjugate it to an amine terminus of our target. Right? And so when you design photoclick experiments, first you need to design a protein or a functional motif that has your molecular plug on it that will be activated by being shown to light. And then you need to design a amine containing molecule that is your target amine. And so then you have the two pieces that can click together. That's what's called photoclick. And so then we have this functional amine, and then usually there's some sort of buffering requirement necessary for the media that you're using it in. And this buffer is going to facilitate water leaving. So this oxygen here is going to end up leaving as water. And you're going to bond the amine terminus here to the amine terminus of your target. And you can conjugate your functional motif to a target of some kind. And it's a highly efficient process, which is really nice. So you don't get a lot, so as long as you design good plugs and targets, you don't get a lot of off-target reaction mechanisms. You don't get a lot of weirdness of like poor functionality where you're only looking at 50% of the proteins labeled. You get a really high yield of conjugation between this plug-in and your target. And it's a very diverse process, right? So we can have just a simple amide linkage that we conjugated onto something, or we can even do things like going to lysine-specific unprotected peptides in the backbones of proteins. So if we know that we have some RNA strand that's being expressed inside of our cell, say it comes from a virus, and we have a motif on that virus that we think we know, we can go ahead and build a activator that's meant to target that RNA strand using lysines and actually start labeling invaders inside of a cell, for example. And then we can also do that same thing with lysines using just protein backbones, right? And the other thing to remember with photoclick is you can use it to be a deactivator as well. Say you want to stop things from activating. Well, design your plug to attack a functional part of a protein, conjugate your plug onto that functional part of the protein, and now you've rendered it inert through the activation of light on your protein. So it's a really cool way to do assays where you want to have a loss of function. And then we can go the large scale, right? We want to label these mitochondria. We can conjugate something onto the mitochondria and tag a mitochondria. Remember, this functional motif here can be a GFP molecule. We can slap a GFP molecule on there and have that go to a different part of the cell and label things for us. Photoclick chemistry. Very cool. Very hard, but very cool. So I wanted to leave you with an actual piece of data that was generated using quantitative microscopy. So this is a study that we published in collaboration with Tao Wen and Martin Wer from Princeton University. And we were interested in the temporal entry of NLS-tagged peptides into the nucleus during embryogenesis. And so we got these peptide fragments, we call the SV40 NLS, conjugated to an M-cherry molecule. And then we threw that into Xenopus extract, and we encapsulated that inside of a droplet. And so you can see at the early time point, before we had a functional nucleus inside of this droplet, there was the diffuse signal of the NLS indicated that there was no localization of our peptide yet. And then we did a time lapse where we just imaged this thing as a function of time, and you can see the nucleus as it sucks up this NLS containing protein starts to grow brighter. And then at 55 minutes, we have a very bright nucleus with a completely dark background, indicating that we have sucked up all our NLS from the cytoplasm. And by comparing the amount of cytoplasmic NLS protein to the nuclear protein, we were able to determine a rate of import of our protein of interest. So we took this M-cherry NLS SV40 as a control, and we compared it to PHF5A-GFP. And we did the exact same assay, where we started out with none localized in the nucleus, and then as a function of a time, more of it moved into the nucleus, and the nucleus got relatively brighter, and the cytoplasm gets dimmer, and we can compare the import rates of these two molecules. We can then take those import rates and compare them back to the observed mass spec binding affinity that was determined for each one of these proteins, and we were able to determine, and the end all be all of this is that we were able to determine that the affinity that a protein has to bind to the carrier molecule, which in this case is important, is what determines the import timing of a protein during embryogenesis. Most of these proteins that you see here are either polymerases, so they're responsible for DNA translation, or they are promoters, so they're important for binding to DNA. So the difference of import timing for these various proteins ultimately ends up telling us something about the way genes are expressed during embryogenesis. And so with that, I hope you guys enjoyed this kind of introductory overview of microscopy. I hope you learned something, and I will see you in the next one.